Ready to get started if everyone wants to come and have a seat. And also want to welcome our audience that's coming in by live stream right now through the Facebook page. And also want to point out that you all have note cards on your table, and if you have questions, be sure to write them down and raise your hand, and we'll have people that can come pick them up. And if you are watching us by live stream, then you can put your comments in on the Facebook page under the comments, and we have someone here who's writing those down who will also run those up to the stage so we can answer questions that come in on the live stream. And we're just so pleased to be back here again this year. My name is Leigh Pate, and this is the second time we have been able to welcome two really terrific presenters on lobular breast cancer, and we're so happy to be able to offer content on lobular breast cancer here at the Northwest Metastatic Breast Cancer Conference. Our first presentation and topic for this breakout session is going to be lobular breast cancer. How is it different than ductal breast cancer, and what does that mean for me as a patient? And then we're going to follow it up after this presentation with really focusing and talking some about the research and what's coming and what's looking promising in lobular research. And then you can stick around for yet another session where we're going to have a Q&A and maybe a little discussion panel about lobular. So we have a lot of really exciting content today on lobular, and we hope everybody can ask a lot of questions and get a lot out of it. I want to introduce our two speakers today. First, we want to welcome Dr. Steffi Oesterreich, who is a professor of pharmacology and chemical biology at the University of Pittsburgh Cancer Institute. And she's the director of education at the Women's Cancer Research Center, which is a collaboration between McGee Women's Research Institute and University of Pittsburgh Cancer Institute. And Steffi is also the chair of the Scientific Advisory Board for the newly formed Lobular Breast Cancer Alliance, and hosted and helped organize the first international symposium on lobular breast cancer in 2016, which was a real catalyst for launching the Lobular Breast Cancer Alliance, and what we hope will be a whole new initiatives and interest in research on lobular. And then we also want to, Steffi's going to be followed by Dr. Hannah Linden. And Dr. Linden is an academic breast cancer oncologist, and she's active in clinical and translational research. And she is also the professor of medicine at the University of Washington and a full member of the Fred Hutch Cancer Research Center. And among the many other things Dr. Linden is doing is she is also the head of a pre-surgical clinical trial here in Seattle to help really understand endocrine therapies and which endocrine therapies might work better for lobular, and she'll be able to tell us more about that. So a reminder that what you're going to hear here today is general, and just make sure when we're asking questions, we're also going to be asking questions that are going to be kind of broadly applicable to people, and we're not going to be talking about personal medical advice. So when you write your questions, make sure to think about them in broad terms so they might benefit everybody. And then we'd like to welcome Dr. Oesterreich.
Thank you.
Laser and forward.
Pointer. This is forward.
That's forward.
Okay.
Yeah.
Hello, everybody. Thanks for coming. So, as Leigh already said, I'm a professor at the University of Pittsburgh at UPMC Hillman Cancer Center. I do want to take 10 seconds at the beginning, if you allow me, to say this is a very sad day for Pittsburgh. I'm not sure if you have seen the news, but it was a very senseless shooting this morning. It's just like everything the opposite from the meeting we have here, which is all about, helping and supporting each other and moving things forward. And yes, I think it was very, very saddening. Anyway, I would like to thank Leigh and LBCA, because a lot of things which are done in my lab are really driven by the interaction with the advocates from LBCA. So yeah, thank you. I think it has been really great. My task as a PhD, as part of the couple here today, is really explain the science behind it. Sometimes I still find it challenging, although I've been trying this now for years to really do it in a more late term. I tried my best, but if something is still unclear, hopefully we can clarify it in the Q&A afterwards. So I think I can get off the microphone. Okay, great. So as I think many of you know, that lobular breast cancer represents only 10 to 15% of all breast cancer, but given the frequency of breast cancer, this is, a large number of breast cancer patients in the US alone. It ranks as the 6th most common cancer if you separate ductal and lobular cancer. And the number of cancers is comparable, of ILC is comparable to those of brain tumors and multiple myeloma. However, despite this frequency, it is clearly understudied as a breast cancer subtype. So this slide is supposed to show really the main difference between ductal and lobular cancer. Ductal and ductal cancer, as a result of the cells sticking to each other, the tumor more frequently grows as a round lump. In contrast, in lobular cancer, the cells do not stick to each other, and we'll come back to this later on, because that's really the main feature of lobular breast cancer. You lose the glue that keeps the cells together. We call it equiterrin, as a protein. And as a result of this, these cells grow more on a string and not so much as a lump, as a lump. Here are the frequency. You can say approximately 10 to 15%, whereas this histological subtype is a majority of all cancers. The tumors are mostly estrogen receptor, progesterone receptor positive, and not very often HER2 positive. I just mentioned this. This is this protein on the cell surface which keeps the cells together, which is lost, which is the hallmark of lobular breast cancer. They don't grow the cells themselves don't grow very fast, so we call them weakly proliferative. The marker to measure this with scientific terms is called Ki-67. They grow on these long sheets, and even within lobular, not each lobular is the same. There are multiple subtypes within lobular breast cancer. Histological variants, and they are shown here on the right side. For example, alveolar, solid. But what I really would like to highlight maybe is pleomorphic, because we start to understand that there are pleomorphic variants which are more aggressive than other lobular breast cancers. And these mixed, some of you might have heard this. The tumor is half ductal and half lobular. Again, we are at the very beginning trying to understand this. This is where I try to summarize clinical features, and this again will be discussed in more detail by Dr. Linden, who's a physician treating patients. So clinically, I already mentioned this, the cells are disc cohesive, they grow in a string. As a result of this, you can imagine the cells interact much more with what we call the tumor microenvironment. So not only with other cancer cells, but with potentially immune cells, fat cells, blood cells, and other just stuff next to tumor cells. They are more frequently multifocal and bilateral. They are more difficult to excise with clear margins. There's some evidence that there are increased rates of mastectomy. The tumor can be larger because they're harder to detect and to define the extent. There are some studies, again, we need more research, but some studies clearly indicating there's a lower response to preoperative chemotherapy. And there's some interesting discussion that maybe the detection by various imaging modalities can be more challenging. And again, Dr. Linden will talk more about this. There are interesting studies with respect to epidemiology. There's a stronger association compared to ductal cancer with late age at first full-term birth. And they are clearly very hormone sensitive. This came out, for example, out of the, or it's shown that the estrogen and progesterone treatment, the association with increases for breast cancer in the hormone replacement therapies are much stronger for lobular cancer compared to ductal. And finally, a large topic of this meeting here, what about recurrence? There are now a few studies showing that there are fewer metastases to visceral organs. So liver and lung are less frequently affected by lobular cancers compared to ductal. However, there are more unique sites, and these unique sites include the ovary, GI tract, peritoneum, but even, not frequently, but even there are cases where the ILC can metastasize to the eye. There's more and more data showing that there are significantly increased of late recurrences. So this is when we look at the tumor coming back 8, 10, 12 years after the primary diagnosis. This is higher in patients with estrogen receptor positive ILC compared to estrogen receptor positive IDC. Until recently, and I think that's why all of us are here, really ILC was understudied. The first big molecular study where we really took the tumors and then tried to find out, okay, what might be different, is something which was published. It's a TCGA, the Cancer Genome Atlas. I was very happy to be part of this. It's published now 3 years ago, where a multidisciplinary or experts in various aspects of genomic, transcriptomics, really looking at the cells in more detail, got together and analyzed ILCs and IDCs. And this is a mistake. I wish it would be that many, but it's not. It's actually 127. So that 27, that's an error here. So we looked at 127 lobulars and 490 ductal tumors. And then looked, okay, what is the difference? The major result coming out is that lobular tumors had a loss of ecdysferin, this protein which is on the surface of the cells, in more than 95% of the lobular tumors. So this is really the first study which, in a very systematic way, showed that almost each lobular cancer, one way or another, has lost ecdysferin. So this clearly defines loss of ecdysferin as the hallmark of lobular tumors. Other important findings are summarized here. These tumors, ILC compared to IDC, is more frequently estrogen receptor positive. So almost each almost each lobular cancer expresses estrogen receptor, the receptor which binds to the female hormone estrogen. For those of you who are familiar with the molecular subtypes, luminal A, they are more frequently luminal A as compared to the IDCs. I don't want to get into this here, but the study also showed that there are differences in mutations in specific genes. I just mentioned a few here. We haven't really understood this. We don't know whether these specific mutations drive unique aspects of lobular cancer progression, development, but there are a couple of groups now working on this. So what does loss of ecdysferin do? So, I tried to draw this here. So these are supposed to symbolize cancer cells from a ductal cancer, and this from a lobular. So basically in the ductal, these cells they attach to each other as a result of having ecdysferin. In lobular cancers, you lose ecdysferin. The overall architecture of the cells somehow changes because suddenly they are not elongated. They are really much smaller, and they're very round. And you can see this here. This is actually, we can do this in tissue culture in the lab. We can just label the surface of the cells. And this is an IDC, and this is an ILC. So you can see they look very different. And this is exactly what they look like in clinical specimens as well. So in addition to being the hallmark, this is also clearly an opportunity for us to think about treatment. Does the loss of Ikaterin present a therapeutic vulnerability in the sense of, do these cells, these ILC cells, become sensitive to specific therapy as a result of loss of Ikaterin? The first study showing that that indeed is the case was just published by a group in London, and this is shown here. So this is really pretty hot off the press. So they did a screen where they compared cells, breast cancer cells, in the absence and presence of Ikaterin, and look whether there are drugs which can specifically kill cells when they do not have Ikaterin. And they identified a gene called ROS1, shown here, Ikaterin ROS1, and they showed that these they call it synthetic lethality. So the cells die when you lose both Ikaterin and ROS1. They showed this in very clever mouse models. It's a mouse model. Don't want to go through this, but just trust me, you give them the drug, and the mice, the tumors shrink. And now, most excitingly, there is a trial ongoing as a result of this study. So patients with ILC are now treated with a drug as part of a clinical trial targeting ROS1. If you want to have more information about this, this is actually on the LBCA website. And maybe we can talk about this in more detail later. So very exciting. I want to show this slide again. I showed this at the beginning. This is just large R, to really reflect again on the main differences between IDC and ILC. Because if you look at this, you can really think of there must be other differences which potentially we can target therapeutically. So again, these cells in the IDC, they all touch each other. In the ILC, they grow in these strings, and then you can see there's stuff in between. So what we and other groups now trying to figure out whether that differential growth pattern could result in unique dependencies which we can target therapeutically. The three specific areas we are very interested in is growth factors. The growth factors are factors which make cells grow, just like the name says. And very often, these growth factors are not produced by the cancer cells, but by cells surrounding the cancer cells. So given that in ILC, these cells are surrounded so much by the tumor microenvironment, we think that this might be something very unique. And indeed, we have some very exciting data on this, and other groups as well, and I'm going to show this to you. The interaction with the immune cells potentially could be different, right? Again, you think you don't have, the cells don't all cluster together, but there's a lot of space for other cells to move in. I'll come back to that. And finally, maybe metabolism is different as well. The way the cells have access to food, to nutrients, and this could be sugar, this could be fat, lipids, etc. Again, from those three differences as a result of the neighborhoods being different, we think we need to study those. For those, for the different growth factor signaling, I'm going to show you three small pieces of data. I am totally aware that for some of those, that is a little bit more complex. I'm going to share this anyway, and I'm happy to either discuss this later on in the Q&A or even after the session is done. If somebody has more questions, we can discuss this in more detail. I didn't want to admit it because I think it is very exciting, and I think potentially this is important for future clinical trials. So the first one of the growth factors is called IGF-1, insulin-like growth factors. There have been clinical trials in the past, and in the past, these clinical trials have not been very successful. However, we now have data. There's a paper recently published in Clinical Cancer Research to show that it seems like the IGF-1 receptor, again, where we have drugs already, it seems to be more active and higher expressed in ILC. So the idea is maybe if you do a trial where you include everybody, the drugs do not show, the trials are not successful. There's no significant improvement. However, if you have a biomarker and you say, well, how about we do this only in a subset, like in ILC, maybe these trials will be more promising. So this is something we're very interested in. Another growth factor receptor is called FGFR4 or FGF fibroblast-like growth factor. Very similar data. We have recently shown that when you take resist cells, lobular cancer cells resistant to hormonal therapy, they have very high expression of FGFR, of these growth factors. And we have seen the same in sequencing clinical specimens. And this, for example, includes metastasis to the ovary. They have very high expression of FGFR4. There are now very recently some drugs out specifically targeting FGFR4, so we are very interested in that. And last but not least, and I'm sure a few of you have heard about HER2 and HER3. In the introduction, I showed that HER2 is not very frequently, that ILCs, primary ILCs, most of the time are HER2 negative. However, with now increased ability to look at metastatic lesions, we think there's actually an enrichment of HER2 and HER3 mutations in metastasis from ILCs. And I'm going to show you 2 pieces of data supporting this. This is a paper which was published by Christine Desmond and Christos Sartorius 2 years ago, where they did a pretty comprehensive analysis of lobular tumors. It included both primary and metastatic. And they found that there is, I'm not sure if you can read this, there are 5.1% of ILCs had mutations in HER2 and 4% mutations in HER3. So this is 8% of ILCs, and this actually included some primaries. We have now done an analysis, actually I did this for this talk, where we looked at there's a recent study published from Memorial Sloan Kettering, where they did panel sequencing on large number of patients which were seen at Memorial Sloan Kettering, and they made the data available. So you can look at your genes of interest and look, okay, is it mutated or not? And we specifically looked at HER2 and HER3 mutations in metastasis from patients with IDC and ILC. And I would like you to take home 2 messages. First, this is a very high rate, right? So 15% of metastatic lesions from ILC had mutations in HER2 or HER3. And the second message is that it's clearly significantly higher in ILC than IDC. And these are large numbers, right? This is not small. So we think this is very meaningful, potentially with therapeutic implication. That's, again, something which we really need to follow up. Together, all these data signal and say that this growth factor receptor signaling, IGF, EGF, HER2, for whatever reason, is increased in ILC. And in the second part of the session, I'm going to come back to this a little bit more, speculating where do we go with respect to research, where do we go with respect to opportunities for clinical trial. Difference in metabolism, same thing like the other slides. I realize that this is quite complex, and I'm just happy if you can take home, the take-home message that we think there are reasons to believe that the metabolism could be different between IDC and ILC. So this is from a paper we recently published. I realized I didn't put the reference on here, but I'm happy to share this later. That you can take cell lines in culture, we can grow them in a plastic dish, and then you can analyze them. You can look at genetics, you can look how do the genes behave, how fast do they grow. The other thing you can do is you can see what do they like to eat, what we really call metabolism. There's a machine, the machine is called Seahorse. I don't know why it's called Seahorse. But anyway, when we take either IDC cell lines or ILC cell lines, when we look at different readouts of use of sugar of these cells, we found that these ILC cell lines do not use sugar as frequently as IDC cell lines. We don't know why that is, but that's the data we found. Does it have implication for, imaging, let's say, for example. We don't know, but it's clearly something which we should think about. At the same time, when we look at gene expression, so what's the molecular changes in lobular tumors, and we list the top 5 molecular differences between IDC and ILC, one of the pathways which seem to be altered and much higher in lobular tumors is lipid metabolism. It's basically, there are different genes and pathways to take up lipids, fats, into cells and to metabolize these lipids. Again, we don't know why that is, but it is something very interesting. We're super interested to follow up. Clearly, some early hints that metabolism of ILC cells is different to metabolism of IDC cells. And last but not least, just a few words on immuno-oncology. I guess these days, there's no single talk on breast cancer without thinking of immune therapy. I'm sure a lot of you have seen the news on the recent trials in triple-negative breast cancer. There's much less out in ER-positive, no matter if it's IDC or ILC. But there are clearly some data, and some data is published, and some is ongoing, to say that maybe in contrast what somebody might expect, but there is a significant subset of patients with ILC where the tumors are what we call, they are hot, they are basically infiltrated with immune cells. It's not everybody, right? And we are at the very, very early stage. It might be 10%, it might be 25%, we don't know. But when we look at expression of immune genes in IDCs or in ILCs, we can group these and specifically in one subtype, in the molecular subtype of luminal A, compare IDCs with ILCs, and then you call these tumors having either in low or in high immune phenotype. You can see that there's a higher percentage of ILCs which have this high immune phenotype. This data was actually recently published. I didn't put the reference on here, but I'm happy to share this with you. Maybe I can just send this to the organizers or LBCA. There was another poster at the San Antonio meeting last December. San Antonio is where the majority of breast cancer investigators and physicians get together. There was a poster from Foundation Medicine where they have a lot of genomic information on IDC and ILCs, and they looked at something some of you might heard is this tumor mutation burden, or TBM. They can basically look how often genes are mutated in these tumors. And they looked for ER-positive IDC and ILC, and they grouped the tumors into low, intermediate, high, or extremely high. And interestingly enough, what was shown on that poster is that ILCs, metastatic ILCs, have a higher percentage of tumors which have a high mutation burden compared to IDC. Again, it's all very, very, very early, but I think the main message from the slide should be we should work on this. We should understand this further. Interesting enough, there's one clinical trial already open. Again, the data, you can find more details on the LBCA website. It's a trial for patients with metastatic ILC where they combine chemotherapy with an immune checkpoint inhibitor. And the trial is open in the Netherlands, I believe. Right? Yes. So the trial is called Gelato. So again, I think stuff is ongoing, and we need to do more. So to summarize, the hallmark of ILC is loss of Vicaterin. The majority of tumors respond very well to hormonal therapy. Almost every tumor is ER positive. However, recurrence can occur, and unfortunately, very often you have these late recurrences. There are recent molecular studies in primary and metastatic ILCs that have identified unique or enriched dependencies, and resulting already in the opening of some ILC-focused trials. I do have some ideas which I would love to discuss on challenges and hurdles for ILC research and some burning questions I think we should study in more detail. But I think that will be in session 2. And I would like to end with, thanking everybody for support and for the people in my lab. And my lab is actually co-joined with my husband, Adrian Leigh. This is our shared group down here, the funders. And again, a pitch to LBCA, which is like the best group of, motivation and friends I can have. And Leigh, especially running LBC. And thanks for all of your attention.
Thanks. Thank you. I'm going to try to not repeat and talk about a little bit of the clinical relevance here and some of the work we're doing here in Seattle. So lobular cancers, as we've talked about, sort of spread like a net. And in that sense, they can really be more stealth. And that can be frustrating. So you see a greater likelihood of some positive margins or a little more likelihood of some surprises after a lumpectomy. And as Steffi went over, they're more likely to be hormone-driven. And they're more, I use a word called indolent. That's just like a low-key 67. They're not growing fast, and that's why they can recur late. So this is my little graphic attempt here. And we've buffed this up with a movie. So this is the pattern of spread. And ductal is on their right, kind of a glom of cells. And the reason that lobulars can be so difficult is they can spread like a net, and they kind of spread along the lining of things. And that can be the deeply internal lining, shall we say. So if we're like a worm and we have a tube that goes from our mouth to the other end, there's the inside, which is where the endoscopy or the colonoscopy is visualizing, but then there's the outside. And lobular tumors tend to focus on the outside, and that is why you can have a negative endoscopy or a negative colonoscopy and still actually have a stricture that's causing your problem. And this is where they can be very problematic. And it's sort of how, by spreading like a net, they can kind of get to your ovaries, which are, of course, a trophic site for hormones. Hormones are present on your ovaries, and so the ER-positive cells find them, and we use a term called drop metastases. But the reason I added this little movie to the slide is that, truthfully, often you can see the tumor as some sort of a caking or thickening, like a lining of the around the lungs, the space we call the pleura. You can see some thickening there. And the way you get the thickening is it's spreading like a net, but it's redundant and it's coming over itself. And so those thickened areas are visible by traditional imaging. But that little edge where it's just a thin net, that's much harder to pick up even with techniques like MRI. So we kind of went over this already, but one of the problems we have with the classification is that they're all not the same. And so you can have mixed ductal, and you can have some more aggressive tumors. And certainly we've seen HER2 positive or triple negative lobulars. But most lobulars are low-grade, classic type, hormone receptor driven. What's interesting, and I think you have to remember, is all the drugs that are coming out for ER-positive tumors are probably going to be great for lobulars. And the subset analysis that I've seen from some of the CDK studies shows that, not surprisingly, those are very endocrine-driven drugs. The CDK4/6 inhibitors synergize with endocrine therapy. So you do get something out of the same therapy that's ER-directed being given to a lobular tumor as with a ductal tumor. And it is true that tumors can change their stripes over time. The incidence of that is low. We say 10 to 15%, but lumped in there is progesterone receptor loss, and that doesn't mean that you're not ER sensitive. So the real incidence of things changing is probably 1 in 10, and it may be less with a lobular tumor. So one of the places that's very challenging for imaging in ductal and lobular is bone, and that's the most common site of metastases. And so there are two types of bone metastases. There's a sclerotic and there are lytic. And lytics are like little punched out holes in the skeleton, and those are harder to see. And so the mistake that gets made is you present with a lytic lesion, and then the bone scan shows a lot of sclerosis later on, and the radiologist says that's hotter, so that means it's progression, and actually that's response. So you have to be careful not to overread bone scans, but it doesn't mean that they aren't useful. Just to define this a little bit, so the linings that we see sometimes with lobular tumors that where the tumor can spread, I sort of describe this as it's like we're shrink wrapped, and there's a lining around your heart, there's a lining around your lung, there's a lining around your intestine, there's that whole omental thing, which is just more saran wrap really, keeping different cavities separate from each other, and unfortunately those are sites that we can see lobular tumors spread. So you just have to look in those areas, but it doesn't mean that you can't detect it by imaging, it means it's a little harder. And they can be tricky, and sometimes you can't appreciate it. So if I see something happening sort of indirectly where it looks like the drainage from the kidney seems a little bit blocked, I have a low threshold to investigate that with actually a urologist, making sure that we don't lose kidney function because the ureter, the tube that goes from the kidney to the bladder, can get a stricture around it, again, from the outside, and that can be fixed by a stent. Not fun, but it can be fixed. Okay. So I think the things that we want to consider, in terms of finding the right treatment for each patient are just, did you get a metastatic biopsy to begin with? There are patients who I occasionally see who've never had one. It's just been inferred. I don't think that's a good idea, because I think you want to know your HER2 status and you want to make sure you still got the same thing you started with. And then I think endocrine therapy is exceedingly helpful here. That's really what we're learning from many of the studies that we've already talked about. And I've actually recycled endocrine therapy. So if you've had a great response to one therapy, then we give you a bunch of different therapies. You can sometimes go back to the original endocrine therapy and actually get a response. But more often now, we're going to get involved with these molecularly targeted agents like everolimus or palbociclib. Because I think they push the response rate quicker and give you a longer duration of disease control. I think what's difficult is you want to figure out how to best monitor your tumor burden, and that can be an indirect measure like a tumor marker, or it can be an imaging study. If you've had a really long response to therapy, and let's say you started with a pleural effusion, a fluid in that lining space around your lung, so not in your lung, kind of on your lung, if you get a really long response, like years later, I'm not sure it's going to come back where it was. You've got nice disease control in that area, and it can sort of pop up, unfortunately, elsewhere. So if you really want to know what's happening, you have to think about looking elsewhere, and you have to pay attention to symptoms. And I am extremely enthusiastic about clinical trials. This is sort of like how male breast cancer patients feel. I mean, male breast cancer is extremely rare. But we've advocated that get included in the female breast cancer studies because they benefit from the same therapies. And I would say the same thing about lobular. Don't think that because you have a lobular tumor, you shouldn't be considered for just the regular clinical trial with the new therapeutic that's out, because I think there are some good anti-estrogen therapeutics that are coming up. So I kept this brief. We're going to talk a little bit more about those trials and other things. Just a shout out to the many areas of support, including the research funding we've had through the University of Washington with our program Project Grant Molecular Imaging and our current UO1. Just decided to keep it short, make it work.
So if we can just stay up, I think, and have a seat and do some questions. Okay, so first question, because this I think might have been some news to some folks, is how does lobular breast cancer present when it metastasizes to the eye?
Oh, yeah, it's a terrible thing. And ductals can do this too. The eye is such a funny thing because it's actually connected directly to your brain, right? It's really just a part of your brain that kind of sticks out. So that is technically a brain metastases, and it can present in multiple ways, but often the most common we see is, visual change and maybe some swelling around the orbit. And we have a tight friendship with the ophthalmology folks to be able to get a look at that. And then the way that gets treated almost uniformly is with radiation. And we have much more targeted radiation now, so we can really control that. And You know, I've seen people lose their vision and get it back with good treatment, but that's not a nice place because it's right next to your brain. And so the tougher piece in there is what about the rest of the brain? And is that you want to call that a brain metastases and think about whole brain radiation, which is a big step. So it's complicated, but the way it presents is really a change in vision, usually in one eye.
So yeah, I'd love to say a few words about this. This is one of those research projects which we actually started as a result of talking to some folks here at the meeting a year ago when people said, wow, this is a unique place. And so we have recently looked into this. I had a medical student in the lab who I said, well, why don't you look through our data registry over the last 20 years and identify patients with eye metastasis, just because it's rare. And so he made a couple of very interesting observations. He identified 45 patients with eye metastasis. The vast majority, there's a significant enrichment for patients with ILC. The vast majority of those come from ILC. There are a few with IDC, but clearly significantly enriched. In contrast to what we expected, a lot of them do not have brain mets. So that is like, you know, a unique, they like to go there independent, even if they don't settle in the brain. And very often it's on both eyes and not only on one. So we now requested the tissue to actually look at this and to try to understand, like you said, maybe it's because the parts of the eye where it goes, maybe there's very high expression of estrogen, but maybe it is something else. And you know, we try to understand it, but it's interesting. It's a small subset, but it's clearly something we should figure out.
And I think we have time for one more question, but never fear, because we have a whole Q&A session coming, and we'll save these. This is a question about the what is understood about the long-term dormancy of lobular breast cancer and this tendency for it to come later?
It's a hard one. You know, there's one of our researchers in the TRP, Cyrus Kajar, looking at these indolent cells that hang out, and it's something that honestly I don't think we understand, but it's a dormant state, and it is why I think that the bone stabilizing drugs are effective in preventing some recurrences. But that's an inference that I don't think we can hang our physiologic therapeutic hat on in terms of like understanding a pathway. But it's also why we're now recommending 10 years of extended therapy to people, is because, you know, if you're going to live a long time, you can live long enough to see this tumor again, potentially, and we can delay that by giving you some extra endocrine therapy. It's really part of why endocrine therapy works. And I think the magic trick is that you take the tamoxifen or aromatase inhibitor for 5 or 10 years, and it's still helping you 20 years later. I mean, those curves continue to separate, and that's amazing to me because you're off the therapy. And I know a lot of people have a hard time stopping. They have a hard time stopping chemotherapy, and they have a hard time stopping endocrine therapy, but the fact is it works, and I think it is pushing more of those dormant cells to stay asleep longer or potentially forever. But this is a hard one. I don't think we understand it.
Yeah, I mean, there was a great talk this morning from Cyrus. You know, it's great. I mean, in part, we don't understand it better because it's very difficult to research. We just don't have models, really good models for dormancy, right? I mean, it's just very hard to study. But folks like Cyrus and others, I think, hopefully will make progress because we understand so little about it, and we need models to study that better.
Well, thank you very much. We're going to take a very short 5-minute break and then come back and pick this back up with what's new and what's coming with research. So thank you.